Hello, everyone, and a very good afternoon to all of you, and a warm welcome to today's webinar entitled Insights on Feluda, the first indigenously developed CRISPR-based SARS-CoV-2 test. The COVID-19 pandemic has required rapid implementation of the known measures as well as simultaneous development of novel tools. Diagnostic has been at the forefront in the disease management, and many of the healthcare professionals who are joining the webinar today have real life experience starting from last March to this March. We are almost a year into the pandemic now. While we were working on the field, scientists and innovators were involved in discovery, development, and inventions to tackle the pandemic. That brings us to the today's topic focus on the Feluda, a CRISPR-based SARS-CoV-2 test. A recently discovered biological phenomena called CRISPR. It is a defense mechanism in bacteria against the viruses that is pathogen, and it can be used for editing of the genes and genomes. Uh, and it has a lot of translational applications. The CRISPR-Cas, so Cas is like CRISPR-associated system, is such an important discovery that this year Chemistry Nobel Prize is awarded to Dr. Emmanuel and Dr. Jennifer for their work on CRISPR. So it's a very important technology, as well as, we have it's a new discovery, and we have a new virus as such. So scientists have definitely been at forefront to bringing all the things together. So today we have Dr. Devjot Chakrabarti with us, who has worked extensively on basic and translational aspects of CRISPR technology. Dr. Chakrabarti has completed his master's in molecular biology from West Bengal and PhD from Max Planck Institute of Molecular Biology and Genetics and Technical University, Dresden. After 2 years of postdoctoral work at Biotechnology Gentra as a medical faculty in Technical University, Dresden, he joined CSIR IGIB, New Delhi as a scientist fellow in 2016. Dr. Devjyoti is presently a senior scientist at CSIR IGIB New Delhi. His lab is basically dealing with RNA biology. It is a vibrant lab of a group of young postdocs, PhDs, and research scholars who share a very deep passion with the science and how we can translate the scientific discovery into people's benefit. The lab is more focused on the CRISPR-based point of care solutions, and we will be having a real life example when he will be talking about SARS-CoV-2 solution, detection solution, and gene editing based treatments for molecular diseases like sickle cell anemia. So his lab works on multiple facets of CRISPR, and probably he will share some of them, but definitely it will focus more towards the SARS-CoV-2. Lab also focuses on understanding neurodevelopmental diseases using organoids. Organoids, you can say like mini brains. Dr. Dev Jyoti was awarded CSIR Young Investigator Award for 2020 and became one of the 30 EMBO Young Professionals, Young Investigators of 2020. So 2020 has a lot of things to do, and some such glorious awards are also awarded this year, last year. In addition to science, he is a renowned sitar player as well, and he is well versed, so multifaceted personality. In today's webinar, Dr. Jyoti will discuss on the CRISPR technology and Feluda test. The webinar will be recorded. I'm sure it's going to be a great learning experience for all of us. If you have any question, please type into Q&A tab. Don't refrain from typing into the chat tab, write it in the Q&A tab. And Dr. Dev Jyoti will like to address as many of them at the end of the session. I request Dr. Dev Jyoti to, Dr. Chakravarti to kindly start the session. Thank you.
Thank you, Dr. Prashant, for the very nice introduction. And yeah, I think more than anything else, we're just one of the second line or third line, I would say, workers who have been trying to contribute in our own ways. And as is a lot of different scientists and healthcare professionals across the world. So, thank you for inviting me to this seminar today and to share some of our insights. It will mostly be centered around the diagnostics that we develop for SARS-CoV-2. But more importantly, I'm probably going to talk today a little bit about what we are currently doing, knowing that the UK variants and the variants of SARS-CoV-2 are around. I think you would find that even more interesting. So I'd like to first share my screen and then you can let me know if all is well so I can start. So just give me 1 second. So can you see my screen?
Yes, it's visible, and
Great.
you are
Okay. Audible.
prominently audible.
Excellent. So, yeah, so I'll get started. So I just changed the title a little bit, um, to emphasize that, CRISPR as a tool, as a technology, was initially brought about as a marvelous technology that could be used to permanently change things in the genome, and therefore addressing the holy grail, that where we are oftentimes considered to be a product of our DNA, and whatever is there in the DNA is there in our destiny. CRISPR kind of shattered that myth a little bit, and now it is possible to actually make changes in the DNA and give rise to conditions that are improved in terms of the health and well-being. So you can correct diseases, and as I say, there are clinical trials for CRISPR editing for diseases that are being carried out in different parts of the world. So um, there are very few people, I think, who have not heard about CRISPR, and it kind of reached a sort of acceptance about the work that was done by Emmanuel Charpentier and Jennifer Doudna with the awarding of the Nobel Prize in Chemistry last year. So the year 2020 was actually a year of women, and I'm sure there's a lot of women in the audience today. Almost 70 to 80% of my lab actually are women scientists, young researchers, and really this year has been championing the cause of women in almost every field that you talk about, whether it is science, whether it is politics, every field that one is, whether it is sports and healthcare as well. And it is really wonderful to see that the entire scientific community came as one last year to award this Nobel Prize in chemistry for the development of a method of genome editing. However, like all scientific discoveries, it has to be kept in mind that any path-breaking discovery is done by work which has been conducted over a long period of time, and when we make such discoveries, we're actually standing on the shoulders of giants. So CRISPR, therefore, is also something that has moved over years, since the 1980s when the first seminal discoveries were made, then a lag phase during the 90s, and then again in the 2000, the first part of this century, when a lot of work went into it. So therefore, CRISPR have edited the two and made it several, because there are indeed several heroes. And if you look at the top row over there, those are the real heroes of CRISPR, because the bacteria and the virus and the archaea, which really brought about CRISPR for our use, oftentimes called simple organisms, oftentimes called not so developed organisms. But I think now with the COVID-19 pandemic and understanding what a virus can do to the world, nobody would dare say that these are simple organisms. So yes, the virus and the bacteria and the archaea, which is where the CRISPR originates, are also CRISPR heroes, because had the sophisticated immune system not existed in these organisms, we would not today be learning about CRISPR and understanding how it can actually be used in the human context. So, what is CRISPR? I think there's a lot of people who are in the audience today, so just trying to bring them all in the same page to understand what CRISPR actually is. It is a type of bacterial adaptive immunity, just like we have immunity, and I think immunity is something that almost everybody around the world has started talking about now with respect to the pandemic and the vaccines. So one of the it's just as a side note, one of the good points about the pandemic, it has been terrible in the way that it has actually spread across the world, but one of the positives to see from it is how science, technology, medicine, healthcare, these are things that are now so well appreciated, and a layman even understand concepts about vaccines and immunity. So that's why I think it's not important to go into details about how the immune system works. What was not known for a long period of time was that the immunity that we often talk about in terms of a human being also exists in a very, very sophisticated manner inside a bacterial cell. So, a bacterial cell, which is shown over here, when it is attacked by a foreign invader, in this case, it's a virus, and you see the virus like shaped like an alien, as you can see on the top left. This virus, which is called a bacteriophage, it actually it introduces its genetic material, which is the DNA or the RNA, inside the bacterial cell. And this genetic material is then recognized by the bacterial cell, and in a very sophisticated manner, a part of that genetic material is introduced into the genome of the bacteria. So it works almost like a librarian, who tags the books that enter the library, so that next time when there's a new person who wants to issue that book, you can use that tag to identify what book has to be issued. So similarly, the bacteria takes a part of that foreign DNA, and it puts it in its own genome at a very specific position, so that it remembers, and it has a memory that this particular infection had happened at an earlier time point. So when the same virus attacks the bacteria once again, as you see from the right corner, on the right top corner, the bacteria quickly assembles a complex of RNA and protein, and it attacks the new invading bacteriophage DNA and chops it off into pieces, thereby resisting an attack. So it's quite fascinating to understand how a simple organism can actually develop such kind of an immune system. To put that into perspective, here is a small video just to tell you how these things happen. So as you can see over here, this is the bacteriophage, which is introducing the DNA inside the bacterial cell. And this double-stranded DNA now enters into the bacterial cell and is recognized by a complex of RNA, which is shown over in red. This belongs to the bacteria, and a protein, which is now shown here. It's called a Cas protein. Now, this Cas protein and RNA, they together form a ribonucleoprotein complex, and specifically tries to look for the signature of the viral DNA from its previous memory. And once it finds that signature by base pairing with the DNA, it makes a precise double-stranded break on that viral DNA, thereby chopping it. Since there are no repair proteins, the viral DNA is therefore chopped, and hence the virus is unable to replicate anymore. Now, scientists were very smart. They realized that you can actually take this information, that you can use a Cas protein and an RNA component to specifically recognize a DNA sequence by putting it inside a human cell. Now, what happens in a human cell is very interesting, is that once in a human cell, this Cas protein goes and recognizes a target DNA, which can be directed by the sequence of the RNA which it carries. Then the similar kind of a double strand break actually happens in the human DNA as well. But of course, in the human cell, since it's an advanced organism, there is a repair protein. There's a repair process which continuously occurs, and it tries to repair the broken ends and pulls them together, thereby sealing off that edge, almost like putting in a fevicol at the broken ends and then just bringing them together. Now, sometimes if you have a broken part of DNA and there is a very tiny template here means a tiny piece of foreign DNA which has some amount of homology or similarity to the sites where the break has happened, then this small piece of foreign DNA can actually be integrated at the breakpoint site, at the break site. And this is what is gene editing. It can be done in different types of cells. It can be done in different formats. It can be done on a single cell level by introducing the stuff, and it can also be used if you have considered that this foreign DNA can be any gene of choice or any DNA of choice. It could also mean that this foreign DNA could code for the corrected version of a disease causing gene. So if you, for example, has a BRCA1 mutation which causes breast cancer, and if you put in a foreign DNA which has the correct version of that BRCA1 gene, then this correct version or the correct DNA template would simply get integrated at the site where the old mutated DNA was. You can understand how powerful that is, and what in terms of applications it can lead to. And that's why repair following the CRISPR mediated DNA breaks is what governs heritable genome editing. Like I said, that if there is a small piece of donor DNA, if you can look on the right side of that schematic, this donor DNA can integrate into a site where the CRISPR has made a double stranded break on the DNA, and then this gets repaired through a process called homology directed repair. An alternate form of repair as well exists, which doesn't oftentimes give rise to very accurate repair. But what we are concerned more about is this HDR, or the homology directed repair, because it essentially allows us to make heritable changes in the DNA of an organism, and this could essentially be a practical solution for genetic diseases where you want to introduce a change at the DNA level. Knowing that it is so powerful, there is actually hardly a field of biology or physics, okay, I mean even in remote areas of sciences where CRISPR has not been applied. And if you look at the timeline from 1980s, when the first rudimentary discoveries were made, till 2020, when you have already started having clinical trials, it has been quite remarkable how many fields CRISPR has been used in. I mean, I can just name a few. For example, CRISPR has been used for trying to bring back the woolly mammoth back to life, by taking out the DNA from the fossils and trying to put it into a different organism. CRISPR has been used to make mosquitoes sterile and then releasing them into the wild so that the mosquito-borne diseases can be actually controlled. CRISPR has been used in diagnostics, like I would talk to you a little bit later. However, probably the biggest and the most important application of CRISPR has been in disease genome editing, where it has been used for correcting disorders, for correcting genetic disorders. And one of the first genetic disorders that are on the way to getting corrected is, of course, sickle cell anemia. In 2006 2019, the first patient in the US who was given a shot of this sickle cell anemia, CRISPR, the CRISPR based editing approach in the hematopoietic stem cells. 1 year down the line, or two, and the paper came out in New England Journal of Medicine last year, is actually doing very well and is not having any aspects of the phenotypes which is concerning the disease in the body of this patient. Victoria Gray is the individual who has received this. So that's like really remarkable how fast this has all moved into the clinic since the discovery of the CRISPR system. However, CRISPR is not just a disease of the West, in the US, but sickle cell anemia is not a disease of the West, but it is there everywhere. And in India, sickle cell anemia is predominantly, present, particularly in belts across different parts of the country. It could be in Chhattisgarh district, in Raipur, in the Maharashtra belt, in the Odisha belt, in the Bengal belt, in different places. Unfortunately, this is a disease really seen more among the rural population where healthcare, our access to healthcare is not there a lot, and all this causes a severe complication because it is a disease in which the blood is now sickled. It doesn't have a circular nature anymore, due to which it is not able to carry as much oxygen as it can, and it leads to episodes of pain, mortality, and so on. Interestingly, this entire disease is caused by a point mutation where glutamic acid is converted to valine in the hemoglobin beta chain. And if you consider it that this is an ideal candidate where a mutation can be corrected using CRISPR, because you can possibly make a change from T back to A if you needed to correct it. So when we started, or when I started my lab in CSR IGIB in 2016, end of 2016, I was working on trying to develop proof of concept correction of the sickle cell anemia mutation in Indian patient-derived stem cells. So to cut a long story short, what we did is that we worked with a different Cas protein than the one which is currently being used, which is Streptococcus pyogenes. Cas9. And this particular Cas protein was from the bacteria called Francisella novicida Cas9, Francisella novicida. And through different experiments, we figured out that this particular Cas protein had some very interesting properties. One of them was that it was highly specific to targeting, so that if you have a DNA substrate which it targets, and you have another substrate which is just a few bases mismatched, then it would not be actually targeting that. So that's very, very important when you consider that you have a protein inside a body for therapeutic correction, and you do not want to take it to another part or of the genome and make a cut over there. So for example, if you in the process of correcting the BRCA1, you do not want to make an change in the p53, for example, that would make the cells cancerous. Similarly, there were some other properties that we found out that were very interesting, and we showed that it could also be used for correcting the sickle cell disease in Indian patient-derived iPSCs. This article came in PNAS in 2019. However, this was more of an academic kind of academic kind of thing. We really wanted to take this to the possibility of having a kind of a clinical trial sometime in India very soon using CRISPR targeting. And I'm happy to say that we have made further progress on this. We have now engineered this Cas protein with our colleagues in Japan, and we are very soon going to start with the preclinical studies to take this ahead for clinical trials. So I won't be talking about that because there are some IP issues there. What I'm going to tell you today about is that realizing that this particular protein is very, very specific in terms of its activity, we actually use this protein for diagnostics, and that's where Feluda comes in. And all this work is actually done with Dr. Shobhik Maiti, who is my colleague, and we worked on this together. Feluda, of course, the name comes from it's called FN-Cas9 editor linked uniform detection assay, and the Feluda essentially is a the naming was given because there were similar technologies that were there for detection of nucleic acids, which were called Sherlock and Detector, which came from Berkeley and MIT. And when we wanted to give a name to our Cas9 based technique, and there were two Bengalis in the group, so it became automatic that we called it Feluda. Of course, this technology was approved by ICMR and DCGI last year, and this was licensed to Tata Medical and Diagnostics, and they are using currently. It's available commercially for different diagnostic purposes. This was there was a very moving tribute that came out also sometime in November, because the character who plays Feluda in Bengali movies and Satyajit Ray movies was Soumitra Chatterjee, and he actually passed away. So that was something that happened also during the same time. So how does Feluda work? So what we are looking at Feluda over here is essentially we are looking at the activity of a Cas protein on a DNA substrate. So when this Cas protein can recognize DNA, it goes and it recognizes very specifically. It binds to the DNA and it cleaves the target. When it is unable to do that because there is a mismatch in the target, which happens when there is a point mutation, let's say, then there is no binding or cleavage that happens. And this entire thing can be adapted to different types of readouts. It could be on a electrophoretic on a gel, on an agarose gel electrophoresis. It could be through a capillary gel electrophoresis. It could be a fluorescence based readout, and as we will show you that it can also be adapted to a lateral flow, which would be on a kind of a binding based readout. And what was also, like I said, that you could modify this Cas protein, put a fluorophore tag on it, and thereby also make it possible to detect using fluorescence. And that's the affinity based Feluda, basically, which essentially can look at the binding of the Cas protein, and then give rise to a kind of readout which tells you whether the mutation is present or not. And we did this also on blinded samples from sickle cell anemia coming from the field, and also samples which were heterozygous, which were carriers of sickle cell anemia. Normally, these kind of samples which are carriers is very hard to detect using normal kind of tests, because and you have to do a sequencing in order to identify them. Sequencing, of course, takes longer periods of time, and it's it's not something that you can do on the field. Understanding that most of these people are coming from the rural population of India. Feluda can do all of these within an hour, essentially. One of the interesting projects that we looked at was actually trying to see whether Feluda can be used for bacterial infections. So here we collaborated with Dr. Govind Makharia at AIMS, and we got Helicobacter pylori samples, which causes gastric ulcer, from the biopsy samples. And there are different versions of this Helicobacter pylori bacteria, which have got point mutations, and based on the presence of these mutations, the bacterial strain is either sensitive or resistant to the first line antibiotic, which is rifampicin. And in most cases, what doctors do is that they take a scrape of the biopsy, get the bacteria, and then they culture it. And after 3 days of culturing, they know whether the patient should be given an antibiotic or not. Using Feluda, all these things can be actually done within an hour, actually, at the OPD. So we are moving, towards how to see how robust this is, how this can actually be done more commonly in the next few months. And before the pandemic started, we really wanted to take this to the field and see whether this can be done right there where it is needed the most. And this was at a tertiary care center in Chhattisgarh district in the Raipur, in a village in Raipur, actually. And normally the government runs camps in these areas where they do something called a solubility test. It's a very crude test to identify if the proteins are aggregating or not, and it gives you some kind of idea whether there is a sickle cell phenotype which is there. And in these camps, you have these children who are queued up to give their blood, because it happens from blood. And as you can see over here, some of these kids, like the one over here who doesn't have slippers on, is not very excited about the prospects of giving the blood to the doctor or the clinician who is sitting over here. So we came up with the idea of conducting Feluda using saliva samples. So we would ask the kid just to spit into a small tube, and it would have the right reagents that would just break open the cells, get the DNA out, and then a Feluda assay can be done very quickly. It's very important to understand that a lot of such patients or lots of such individuals who come to these camps are very difficult to track at a later point of time. By the time when sequencing has been completed, let's say in a city which is 1500 kilometers away, and results have come after a few weeks or months, sometimes these individuals are actually not even, it's not possible to track them as well, because there's also stigma which is associated with certain diseases like sickle cell anemia. And thereby it makes it very useful to have a technology or a point of care solution where you could just have the patient or the individual sitting there, and the technology can actually detect it. So that's where we wanted to go to. But of course, then the COVID-19 pandemic struck, and we identified a way to take all this whole platform into an assay where we could use it for detecting the SARS-CoV-2. And I think this has been there in the media quite a bit. I'll explain it once more. I would emphasize that it's with all the attention that it got, it's just one of the different types of techniques that people across the world have put forward. And there are several such very nice techniques that have also come from India as well. So it's a kind, sometimes I feel it's kind of unjustified to have so much attention just on this, but there has been a lot of nice developments in diagnostics that have come. So Feluda, of course, is one of such diagnostics, which is CRISPR based, and essentially what it does is the sample collection is very similar to that of RT-PCR, where the sample is collected from the swab or the from the nasopharyngeal, oropharyngeal swabs, or the saliva, and then the RNA is extracted. And once the RNA is extracted, there is a round of reverse transcription PCR or an RT-PCR, which happens in a normal PCR machine, not a quantitative real-time PCR machine. So once you do that on a real-time on a normal PCR machine, which is a 1/20th the cost of the real-time PCR machines. So you can almost think that this can be scaled up 20 times at places where real-time PCR doesn't exist. And then the reverse transcription is done with primers which are having biotin labels, and the amplicon is now biotin labeled. And then there is a CRISPR based association to identify if the 20 bases belongs to the SARS-CoV-2 gene. And combining that with paper strip chemistry, you can have a readout which tells whether a sample is positive or a negative. A negative sample will not have a band at the test region, whereas a positive sample will have a test band, and that tells you whether the sample is actually positive. Just to expand that a little bit, you have the Cas9 complex, which specifically interacts with the target, and the Cas9 is directed to find whether the SARS-CoV-2 signature is there in that target, and if it finds, then it moves along with nanoparticles up into a region which is called the test band. And in that test band region, you have the streptavidin and biotin interaction, so that the substrate is captured. This is how it looks like. So a positive sample or a positive control will have a band at the test band. There is also additionally a control band which is present in every strip, which basically tells if the experiment has worked fine, and thereby can tell you whether this whole thing process has worked nicely or not. If you are a Bengali and you know about Feluda stories, you also know that Feluda is generally assisted by two individuals called Topshe, who is his cousin, and Jatayu, who is a friend in all the stories. And it would be a sacrilege if our Feluda didn't have these two individuals as well. So we had a Topshe, which is a smartphone app that was co-developed with Adiuvo in a startup in Chennai, which basically takes a picture of that strip and then gives a readout telling whether there is a positive or a negative signal. So the biasness of the operator kind of goes away. And we also developed a web tool called Jatayu, which allows you to design primers, and the entire Feluda assay can be designed for not just for COVID, but for any application that you might be wanting to do this. Of course, all this is happens very fast, and you have now a version of Feluda where this can all be done within 45 minutes, and which can also be now done without the need of a PCR machine, which is the isothermal amplification, where you would need just a heating block or some kind of a small dry bath or a water bath using RPA, or recombinant polymerase amplification, and you can get all this within half an hour or so. And the students in the lab also came out with ideas where they would not even need that heating block, but rather use the body temperature. This was more when we were trying to move towards a point of care, like if possible to do Feluda at home or so. You can have all these things pipetted without the need. You can just simply flick them, no need for centrifugation, and then of course put it inside the elbow, and the body heat that is there, it actually does the PCR within 10 minutes, and then one can put them the buffer and the lateral flow strips, and then have a readout at some point. So yeah, so these kind of things we were all developing over time. And the whole idea was that how to make testing so rampant that it can actually help people to come back to schools, to come back to offices, and so on, using simpler and simpler methodologies. But of course, one of the things that happened during the pandemic is that the virus also showed how smart it was. So it also started to evolve, right? And these are what is known as variants. And I think many people have heard about variants already. These are mutations that the virus has picked up. So bear in mind that viral mutations are absolutely normal. As the virus progresses through individuals, it starts picking these mutations up. And some of these mutations can actually make the virus more resistant to vaccines, let's say, or more resistant to any kind of treatment. They can also make it more transmissible, such as those from the South Africa and so on. So some of these mutants are, yes, associated with higher viral loads, and they also sometimes can affect vaccine efficacy. And a lot of places are on high alert, particularly in the USA and the Europe. As you see over here, between from April to February 21, different lineages have started and so on, which makes it very difficult to actually understand in which direction these variations are going. Now we came up with the idea that we have these novel variants. Unfortunately, the gold standard test, which is the real time PCR test, relies on a probe called TACMAN to bind to a certain region in a PCR amplicon, which is specific to the virus, and tell whether there is a presence of the viral signature. But it is not so specific to point mismatches. So it's not a straightforward way by which you can use a quantitative PCR to identify point mismatches or single nucleotide changes on the DNA, which is what a variant actually consists of. Uh, so therefore most countries have resorted to sequencing. Now sequencing has its own drawbacks because sequencing typically takes 4 to 5 days to complete. It requires large data space storage. It's an expensive affair. It is not a point of care diagnostic. So we realized that the FN-Cas9 protein, like I showed you earlier, has a very high specificity to even point mismatches. And here we are actually dealing with point mismatches in the form of variants. So what we came up with is called RAY, or a rapid variant assay. It was almost normal that if the Feluda would be able to identify the COVID signature, RAY, which would be a tribute to the creator of Feluda actually, would be able to identify even point mismatches in that signature. So what it does is that it uses this high specificity of FN-Cas9 to specifically recognize the point mismatch and give a kind of a readout, which is has by because the it has a universal discrimination mechanism which is independent of target. And we showed this for the N501Y, which is a common variant mutation, which is associated with almost all the three lineages: the UK, the Brazilian, and the South African variants. And using a particular guide RNA which has got a point mismatch, you can see it over here in the cartoon, shown in yellow. You can specifically detect both the SARS-CoV region using a different guide and the N501Y with a different guide, and then you have a readout which is so robust that it works every single time. So that's what you see over here. So if the patient is negative, then there is no signal on the strip. If it's just COVID positive but not variant, then you will have a single one of the guides which is working, which is for the wild type variant of the COVID genome. And if both the mutations I mean, if the mutation is there, that is N501Y, then you would have this kind of a situation, which is what you see in this. And we also tested this with the first samples that came into Delhi in the early January from return people who were returning from UK. These were then validated using nanopore sequencing by IJB. Also has a mobile lab for doing nanopore sequencing at the airport, and we realized that we could actually pick up these samples very nicely, and across a different range of CT values. So this tells us that you can actually have Ray working on this mutation. But most importantly, one is to understand that it is independent of the 501Y. So we have now successfully also designed it for the more dangerous, if one can put it that way, E484K mutation as well, and it can also work in terms of distinguishing that as well. So we have a correspondence in Lancet which is in press, which basically talks about how this can actually help in trying to detect early COVID variants. The idea is that if you have a first pass screening where you can identify at a very very low resolution whether a signature of that variant is there, then you do not need to actually put all your samples through for sequencing, because that's not possible. That's very 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 difficult to do that, both in terms of time and cost, and also the logistics, where you would have to quarantine people for 4 or 5 days to identify if they have got a variant signature positive or not. This whole process takes 75 minutes. Now we can do this within an hour, and of course the cost is several magnitude lesser than that of deep sequencing. So the scientific part of the talk actually ends. I have a few slides over here, which basically is telling you that as we move from an idea to a product, what are some of the lessons that we learned. Of course, one of the things was that it had a lot of impact in terms of media attention, because it's one of the first CRISPR tests for COVID which got commercialized, and the approvals came last year. And just to tell you that this was something that was really moved very fast, and we had the idea for this sometime in January 28th, and the working prototype by March 2nd, the patent in March as well, and the technology licensing to Tata Sons actually, and then they formed a new company around this, which is called Tata Medical and Diagnostics, and the company then got an approval in September for market release. So normally, ideas to product, they normally take a much longer period of time. So one of the things that we learned is that during the pandemic, things move pretty fast if one has a very dedicated ecosystem which supports it. And just to tell you that this is indeed true, this is a screenshot of the WhatsApp group which we formed in January 28th, even before the first case of coronavirus was actually detected in India. And then sometime in March, with one of the first samples that came to Delhi from NCDC, we could actually show that this thing was working. You can see the control band developing, and now the test band coming down there somewhere. And of course, in September, the test was approved. So along the way, we learned a few things. One of the things is that we have a different ways of having discussions between scientists and people who are from the industry, and the industry requirements are quite different. The scientific ideas are quite different, and it oftentimes requires interpreters and understanding how a test actually needs to behave to have maximum coverage. And over iterations, I think this number is very old. We have now done almost more than 3,000 4,000 tests, and we're still doing this in terms of improving the product. We realize that a large number of things depend upon the quality of reagents, the quality of RNA, and most importantly, policies as well. Because when you have the market flooded with qPCR kits, it's very difficult to really understand what do you consider as a gold standard in order to compare your testing. So all these things we learned during the whole process. It's a very valuable learning experience for the whole team. The second and the most important aspect was that our students, which is the workhorse of the research labs across the country, they are our biggest asset. And this is a group of people, and it's an expanding group of people. Actually, more people joining there. They really are the ones who built this entire protocol, the test, the assay, whatever you call it, within a timeline where people were just simply going into lockdown and taking off. So these individuals stayed back in the labs. They worked continuously, tirelessly over days and months, and several of them also turning COVID positive at some point of time, and still at it, and they are still at it, trying to identify better and better ways of identifying the variants and so on. I put this slide also across because there are sometimes people who are also from the funding agencies and from the from the different sources which gives fellowships and so on, to understand that one of the reasons we oftentimes complain about people leaving academia or leaving young students who are going out, is because we are sometimes not taking care of them enough. Very basic things such as releasing fellowships on time. So during a pandemic, I think the strength of this entire student force and this young researchers came into the forefront. And I think it is a lesson for us also that we need to sometimes think about being able to take care of them even better. So they say that raising a child requires a village. If you have to bring a product to market in this kind of a time frame, it requires a whole organization. So right from the person who delivers the reagents from the airport to the lab, to the highest possible office, whether it's the principal scientific advisor to the PM or the DG or the director, they all came together in this particular Feluda process. And I mean, the number of people I have to thank is just endless. So many people who were involved. Right from the beginning, social media was taking a lot of attention. There was a lot of attention about it, and it sometimes can go the other way around. It can be cruel, and people were getting impatient and so on. Not real. Sometimes it's difficult to explain to people that when you have a new technology and you have to start from scratch, right from setting it up to validating, to building and producing it and testing it and in the field. It which what takes years, and to push all that into a few months, it sometimes can cause delays. But of course, when things work out and you have social media which make science accessible to public like no one else, it is really very heartening. And one of the very nice things that happened was in September, when Feluda was actually trending after Sri Krishna and press freedom in our country in Twitter, and that was very very nice to see. And of course, Shashi Tharoor, tweeting about it as well without using any of his bombastic words. So that's a good feeling to have. It tells you that one of the things that the pandemic has done is that it has brought science closer to people like never before. And I think everybody is now aware of testing. Everybody is aware what an RT-PCR is, antibody is, antigen is, and of course now vaccines. So with that, I'd like to end my talk. Like I said, there's a long list of people. We have a huge lab, as you can see over here, not fitting in one table. Very generously funded by a lot of different funding agencies. Tata Medical and Diagnostics, of course, has been a partner for the Feluda process and is still one. And yes, if you have any questions, I would be very happy to take them now.
That was an excellent talk, Dr. Dev Jyoti. And actually, I mean, it went beyond like, let's say, detection of SARS-CoV-2, but that was very, very, informative in other disease area as well. In fact, I was planning to have a question on personalized medicine, role of CRISPR in personalized medicine, but your own example of sickle cell anemia gives tells a lot about that. So coming back to the questions which are over here, most of the questions are more towards, some basic concepts like what is the sensitivity and specificity, what are the genes which is getting covered in, especially in the case of SARS-CoV-2. So can you please give your opinion on that? I understand sensitivity and specificity. Your question is compared to what when you are
Right.
not very sure on which RT-PCR, but definitely these are the major
Yeah.
questions which I see. Yeah.
Yeah, so I think a number of them have come up across sensitivity, specificity. So at the lab level, the sensitivity and specificity was somewhere around 97% to and 100% respectively. For the commercial kit in the that is coming from Tata Medical and Diagnostics, this was close to 96 point something and 98 point something. Essentially anything which is above 95% is within the range of almost close to 100, basically, because you would have one day it comes up at 100%, another at 99%, because it's compared to also another test, which is the real-time PCR test. So that is for the sensitivity, specificity. Do you want to take the questions, or should I just go and look at them myself?
You can look on that, but the questions are like, okay, so what is the gene covered over here?
So the Tata MD check kit, which is the commercial name of Feluda, actually
Yeah.
looks at the S gene, the spike protein, and it
Okay.
looks at the region in the spike protein, which is of course not undergoing mutations. We have almost on a weekly basis we look for regions or that region to ensure that there is no mutation that is happening in those regions. And yeah, but we also in our at the lab we also had another gene, which is the N gene, which was also targeted. Yeah. So we
Uh,
tested this for approximately, I think, 22 different regions of the SARS-CoV genome, and identified 2 to 3 regions where there was the highest robustness in terms of detection.
okay, that's good. And when we are comparing with, let's say, that's a very promising thing that you can differentiate into, tyrosine, arginine differences, and 501 is being very critical. In the sequencing, one drawback is like, okay, you have to have a good amount of RNA to start with. So how does it compare when we are talking about variant calling, especially when, let's say, it is, early stage of the disease or late stage of the disease when the RNA quantity is pretty low?
That's a very good point. Actually, sequencing is limited by the amount of RNA that goes in. Feluda, of course, is not dependent on that, because it goes down to detecting a few tenths of copies of the RNA. So therefore, definitely there is an edge over sequencing. What we have not yet done is that we have not yet, for the variants particularly, we have not really gone down to extremely high CT values. Is something that we are currently doing, as we try to develop something that can actually be used as a diagnostics for variant calling. But for the standard Feluda, which works for detecting SARS-CoV-2, one can go down to I'll go as high CT values as 38, 39, or 40, which are sometimes in several places not even considered as I mean, it's well known now that anything above CTs of 30 to 33 are clinically not spreading anymore.
That's pretty good. If it is like tens of copies, then we are talking about really some a very good limit of detection.
Right.
And another set of questions, if I go through, I mean, they fall into the standard diagnostic procedures, like to make it, a rapid deployment. There are a couple of points like the quality we have already discussed about sensitivity. What about the
Mhm.
cost effectiveness? In a sense, terms of affordability, it cannot be the exact cost. Ease of operation and scalability, because those are the important criteria when something is to be transformed from, let's say, lab to the bedside, or into the field, or let's say in the Kumbh Mela or something like that, where you have to screen thousands of people, and we have lot of such gatherings. And here, we talk about antigen test, which is the sensitivity is comparatively lower, though specificity is very high, and their Feluda type of assays can work much better. So three particular criteria, let's say, scalability,
So I
affordability, and
think,
ease of operation.
yeah. So I'll answer regarding the costing. So the final cost to the consumer is of course not something that as an academic we have much let's say we have any control over, because it is governed by multiple factors, which is sometimes it's also the price which is capped by the government at a given place, right to the imports and everything
No,
that
it's
the.
a relative cost. Definitely, I'm not talking about the cost, but it's
Yeah. I
affordability,
will,
I will say,
I
rather
will give
than the
you
cost.
an
Yeah.
idea that the major cost at this moment seems to be the paper strip, which is the major component, which is the cost of the, and we're trying to make it completely indigenized so that this cost can also come down. But of course, one has to realize that RT-PCR costs have also come down drastically over the last few months or so, not necessarily correlating with very good quality. So the Feluda test was actually built to make it either similar in reagent cost or even lower than the RT-PCR costs. But we do see that even at the level of RT-PCR kits, which are kind of now flooding the market with different types of kits, some good, some bad, some intermediate. The costing is definitely it's a very very tricky situation, to put it that way. But again, this is not something that I'm very well aware of. It's something that probably
Yeah, I
the
understand.
commercial
Mhm.
partner would be able to give more ideas
Yeah,
on it.
definitely, completely understood. Yeah. And what about scalability and ease of operation in comparison to when, let's say, you have to deploy at a large stage?
So again, there are a lot of ways by which I think the company is trying to make it. I have heard that there are possibilities of, and they even have possibilities of now having this end-to-end automated with no human intervention as well. A lot of things are possible. So indeed, it's a normal PCR machine, so it can be taken to places where the real-time PCR is not there. So it can be bundled into a small mobile van, and it can go around in the airport. It couldn't be at the Kumbh Mela if it is not already there. Again, something that I have heard about, but I am not very sure, so I'm not talking about it. But yes, there are a lot of different places where it is being promoted. Yeah.
Okay. Is there any alternative, in a sense, like rather than PCR, you're talking about isothermal amplification, like TMA or other things, which can
Yeah.
make it faster and then convert it to cDNA level at the end and, quickly do it? I mean, I don't know, it's a idea or thought process or
So
technically how it
we
works.
started working on something where one could actually do this directly from RNA without the need for a cDNA or a PCR step in between. We have some preliminary data showing that in principle this can work. The complication is actually the sensitivity, because if you do not have an amplification step, then you are working with the true numbers, and that is then a question about how sensitive your detection device is to capture it over the noise. So I would rather go for a signal amplification and accurately classify a sample rather than missing out due to train the algorithm, or let's say train the detection system for that. So Yeah, so I think from
Yeah,
directly
definitely.
RNA, but it can have other applications. So we are looking at that closely. Yeah.
Uh, I believe we can have a look on the questions if there is something important missing over here.
Yes, so I think I'll quickly go through them. So, what is
Yeah,
assisting the donor DNA to ligate at the targeted site? The donor DNA is getting ligated by a complex machinery of proteins. These are all recombinase based proteins, and there are a number of them actually. So, it's a well studied. It's a HDR pathway has been studied a lot, and there are a number of proteins which help it. Second one addressed, can you share paper where CRISPR-based test was compared with PCR? If you're talking about our CRISPR-based test, yes, there are at least now, two papers which are there on medRxiv. They're all in review, so you can look them up in medRxiv, and of course it has been tested, compared with RT-PCR, because that's the gold standard. Then there's a comment. Then there is Sachin Kumar who is asking, can we generate a strip to detect AES causing viruses? I'm not very sure what AES, but if it's a disease, then yes, it can definitely be done.
yeah, it's like encephalitis, I believe he's
It's
speaking about.
encephalitis. Okay,
The
so
acute
then definitely
encephalitis
yes.
syndrome. Yeah.
Yeah, it has nothing specific to COVID. So as long as there are 20 bases that is specific to AES, it can detect. What SARS-CoV-2 genes? Like I said, it detects the S gene, it detects the N2, and we have also other panel of other some guide RNAs which can also be used if required. Same VTM as RT-PCR? Yes, absolutely the same VTM because the extraction process is same. Has the test been compared to culture? I'm not very sure what that means. If if somebody can explain that a little bit later, Vinay, if you could type in what you want to know. Then there is, what is the sensitivity specific limit of detection? I think we have already answered this. These are more than 95%, and about 100 copies of the starting RNA very accurately it can be used. Next question is also the same. No clinical data presented. These are all clinical data actually. So they are all coming from patients. So these are, none of them are synthetic, these things. So there are, I think at our end we have close to 3000, 4000, 5000 samples, and I think similar kind of numbers also the Tata MDL is also pursuing, or even more actually. So, do we currently have CRISPR Feluda available? Not commercially. We are looking at how to get that working. There's also interest from the World Health Organization as well. So we're seeing how to move that. And do we need a different kit to identify each? Very interesting question here, since we are actually identifying a variant. So it is not a replacement of sequencing, because sequencing is generating the whole sequence of the virus. So it not only tells you about 501, but it also tells you about other variants. Feluda cannot be done in that mode. Feluda can identify a single signature variant. So yes, you would need a different kit for identifying each mutant variant. But if you look at the list of the variants and the lineages, you would realize that not all of them are phenotypically giving rise to a complicated problem. So there are very specific signatures, such as, like I said, the 501Y, which is common across the three lineages, and more specifically the E484K, which is causing higher transmissibility. So the idea is that if you can screen for these out, then you save on time and cost for doing a kind of benchmark sequencing. Shruti Shyamsundas, can you tell us about future versions? Yes, there is a lot of future versions which is in the pipe, which is there in the pipeline. Actually, we plan to make panels for diseases. We also plan to take this into a format where you do not need the strip, but maybe through a very small handheld equipment, be able to detect it. But that's, I think, another separate story. What do you mean by interpreters? Oh, I think it's coming from that slide. By interpreters, I mean people who can understand both the aspects of the commercial part of invention, as well as the scientific invention. In our case, we had very good interpreters. We had interpreters who were trying to keep us all sane during a time when we really had to move fast. The cost of the test, well, the cost of the test, at least in the if you look up the government e-marketplace, it is somewhere around 500 rupees. But I'm not sure if that's the current cost, so one can check this. It's This is the cost that I had seen a few months back on the gem platform, the government e-marketplace. Any sensitivity specificity changes, very important question. In different variants we are doing that now, so to check if sensitivity specificity remains the same. A limit of detection, I think I have already addressed this. Till 100 copies we can very very accurately detect it. At a CT value level, this is about 38 to 39, where it can very accurately detect, I think, close to 40. It's probably just about a few copies. How does it terms of cost? Rapid antigen RT-PCR. I think this is not something that I can really tell. There are very good RT-PCR kits which cost few hundreds, and there are, well, some kits which cost about 40 rupees. So you can compare between them. How do you have an enzyme at that kind of a cost? Is it a screening or a confirmatory? So according to DCGI and ICMR guidelines, it is now a confirmatory test. So if you do a Feluda test, you do not need to have an RT-PCR confirmation. Both the genes are targeted in the same test. No, it's not multiplexed. At one point of time, you can amplify one product and then you can run that. So S and N have two different strips. How many viral copies are detected? So I think this I have already answered. There's also IJB team working on TB AMR, where hands are pretty full. But we'll see how to go about this. Either, individually, we have a lot to do at the moment. So I'm not sure how many diseases we can tackle, but we will see how to find a way to go for more diseases. PhD thesis. I think there's a PhD program which IJB runs. You can find the details in the on the IJB website. So isothermal based amplification, true POC, very good question. We know that it works using RT-RPA, recombinant polymerase amplification. But sometimes I think Kavi Ramjit asked this. RT-RPA is a technology which is has a very strong patent portfolio associated with it. So even if we can get it to work in the lab, to be actually be able to deploy it as a commercial test requires a lot of background work in terms of understanding of the patent laws. So I think this is something left to the company to figure out how, and I'm sure they're also working along those lines to see how to actually deploy it. Again, Ravinder Kaur asks, is it able to screen each and every COVID variant? Very good question. At the moment, with the current version of the Cas protein, we can target, I think, 11 out of 21 of the mutations. With an advanced version, which we have not talked about today because of IP, we can target 17 out of the 21. But like I said, that not all need to be detected as a diagnostic technique. You only need to find out the signature sequences. Differences, the one for strain typing. Does routine Feluda testing kit incorporate signature testing strips as well as to identify different genome? Or there are two different kits, one for identification and for strain. So the Ray platform can be combined with the Feluda platform into one single platform. Alternatively, a test which is positive for RT-PCR or a sample can go into the Ray platform to identify whether a mutation is there or not. So both the options are there. Oh yes, saliva sample, somebody has asked. It's easier and cheaper to use. I'm not very sure if there is the approval for doing that, but yes, we have done it with saliva samples, and indeed it can be easier and cheaper to use this sample. For what all diagnostic diseases? At the moment, only SARS-CoV-2. We have not yet had the time to really go for other diagnostic disease validation. Why the S protein is chosen? Very good question. S protein has a larger number of transcripts, so it's a unequally transcribed versus the N. So therefore, it has a higher sensitivity if you design your guides against the S protein. We need to take, yes, the same safety precautions like an RT-PCR. Of course, if in an automated, done in an automated format, all these things will probably be inside that automated chamber. Are preparing a new test kit for multiple virus or bacteria, as in multiplex PCR biofire kit. I'm not very sure what this question is, so maybe you can type it once more. Can you do multiplexing with Feluda? We have not yet tried, but in principle, it would require some optimization, but can be doable. Diagnosis of tuberculosis, yes, it can be. If there was a light colored band, you consider it positive or negative. So again, a very good question from Dhruti. So the Feluda works on the principle of Cas9 binding to the target. So if there, it's a total affinity based as opposed to a collateral cleavage. So if there are lesser number of targets, you would have lesser binding. If you have more number of targets, you have more binding, more nanoparticle accumulation, stronger band. So the light colored band generally happens with higher CT values, and around 35, 36, this is really, really faint. Hence, we have actually developed this app, which is called Topsha, and I think the Tata MD Check Kit also comes with its own app, which is trained by which using artificial intelligence to over a large number of samples to actually categorize whether such samples should be clinically positive or negative. I think it's very important to understand that, when we do an RT-PCR test and we find a value, we oftentimes we see that whether this value is a positive, as a classification of whether a sample is positive or negative. In reality, a lot of things can give rise to values, and just like in RT-PCR, even if you have a background amplification, that might give rise to a qPCR CT value. Similarly, a very light colored band could either mean there are very few copies of the COVID RNA, or it could be just a kind of background, because some nanoparticles erroneously came over there, which is why an app actually helps in identifying such kind of really borderline cases. Can we use this study for DNA? This is actually done on DNA, so it's RNA transcribed, RT done into this. Richa has asked how indigenous is the kit. By indigenous, so I think there is no technology which is completely developed using a single lab's expertise. So we have been inspired definitely by similar CRISPR technologies, which uses Cas12 and Cas13. Of course, Cas12 and Cas13 uses collateral cleavage, and it works on the principle of cleaving of reporter molecules. FN-Cas9 has its own mechanism of DNA binding, which is highly specific, and we are using the binding or the affinity based detection for doing that based on the work that we did over the last 2-3 years. Can we use any type of body fluids as sample? Typically, yes, as long as there is DNA in it. Biosafety, I think we have addressed. Any different saliva NP? Saliva generally comes up at a slightly higher CT values in an RT-PCR, and a lower, intensity band for Feluda. Can influenza and COVID be detected? Technically, yes, can be done. So did you find concordance with saliva and NP or of swabs? Like I said, swab has typically always given a more sensitive reading than saliva. What is the storage condition test? I think standard, these are enzymes, so they have to be stored appropriately. Proteins and enzymes, -20 would be the one. If light color band, then should we repeat the test? Um, I think one. I'm not sure what the so you should go with what the app is suggesting, because this has been trained for that purpose. So if the app is saying that there is a repeat required because it just lies at the threshold, as you would have if you have an RT-PCR which is positive for one gene, let's say, or a pan-Sarbeco, then you repeat. One can repeat the test. Yes. How can we assess the severity? I think clinical decisions are probably not made using diagnostics, so I would not go for that. Can target labeled with biotin saturate all the streptavidin leading to false results in lateral flow? A very interesting question. So can target labeled with biotin saturate all the streptavidin? No, it doesn't actually, because the nanoparticles are only recognizing something called a fluorescent amidite, and this fluorescent amidite is put on the RNP complex at the 3' end of the guide RNA. So the nanoparticle first latches on to the FAM label on the guide RNA, taking up all the RNP complexes up. So if your biotin amplicon is not bound by Cas9, which it will not be if the target SARS-CoV signature is not there, then it will not be actually going there and giving a band. So that's why in every test we have something called a no template control, to understand whether just the biotin label primers, which also go into that no template control, is giving rise to any kind of band. And we are now very, very confident that this doesn't give any kind of background. So I think that covers almost everything. If you have anything else, please do let me know.
That's a great session, and I think almost 44 questions, back to back answering. And probably we are also crossing the timeline. It's already 5:11. You might be delayed for your next assignments. So with that, I believe most of the questions are answered, and I mean we can look into ending of the session. It was actually a great question and answer session, enriching the learning and experience. Uh, we take this opportunity to sincerely thank Dr. Chakrabarti for his informative session. We also thank all the participants for engaging and for all these good questions which are answered back to back. We sincerely thank Bill and Melinda Gates Foundation for supporting us in this endeavor so that we can conduct the webinars and programs routinely. Hope today's session and value added value to your knowledge and lab atmosphere. We shall keep you updated on our upcoming webinars as and when it is happening, the next webinar. Until then, please be healthy, please be safe. The situation is still improving, but we got to be cautious. Wish you all the very good weekend. Thank you very much.
Thank you.
Thank you, Dr. Devjyoti. Thanks for your time.
Thank you. Thank you. Bye.
Bye. Bye, everyone. Thank you.